Hello, so my name's Clare Turnbull and I'm presenting from the Institute of Cancer Research, and it gives me great pleasure to present our CANGEN-CANVAR programme on behalf of all my co-investigators and collaborators. So cancer susceptibility genetics is changing rapidly. Clearly, the challenges of treating patients with advanced cancer using targeted drugs has become increasingly evident, and there has been renewed focus on screening, prevention and early detection as a really key strategy for cancer control. And logically, of course, you will get greatest impact with minimal economic and clinical cost if you can target those interventions to individuals at highest a priori risk. And we all know we've seen a dramatic expansion over the last 5 to 10 years in clinical testing for cancer susceptibility genes. We used to just have familial cancer clinics with high-risk families. We've moved much more to so-called sort of mainstreaming, testing at point of diagnosis in mainstream oncology, increasing pilots of population testing for cancer susceptibility genes, and a lot of that and other testing being delivered by direct to consumer testing companies. And clearly we have a number of impactful interventions within cancer susceptibility genetics, which is not necessarily true in other areas of genetics. We have screening, so we can offer increased screening or screening using different modalities or different frequencies to those at higher risk. We have surgical preventative surgery and chemo prophylaxis. And certainly the UK has been somewhere where there is great opportunity in advancing our delivery of cancer susceptibility genetics. We have, through the National Health Service, we have a national network of molecular diagnostic laboratories, and these are paired with and work closely with their clinical genetic department counterparts. We also have national data collections. We're very good at collecting cancer data via the Cancer Registration Service, currently held at Public Health England, collecting hospital episode data via NHS Digital, and then data on death is robustly collected via the Office for National Statistics. We also, in the UK, have very strong professional organisations. So we have the BSGM, the British Society for Genomic Medicine, within which we have the Cancer Genetics Group, and we also have the Association of Clinical Genomic Scientists, and these groups actually work closely together, the diagnostic scientists and the clinicians, to really offer joined up care. And particularly in England and the UK, I know elsewhere too, we have seen a really high level of political focus on genomics, really real enthusiasm for leveraging benefit in terms of the improved outcomes in healthcare that might be leveraged. So certainly within England and the UK, there have been a series of white papers over the last 20 years, really emphasising the importance of genomics. In 2013, the 100,000 Genomes Project, large amounts of funding as part of the Olympic legacy. The NHS plan in 2015 was all about personalised medicine, really a lot of it around genomics. The Chief Medical Officer, Sally Davis's annual report in 2016, all focused on genomics, called Generation Genome. 2018, as the 100,000 Genomes Project was near completion, this delivery of the NHS Genomic Medicine Service with a lot of disruption and transformation, a national testing directory, real rearrangement of the labs and clinical services. 2019, delivery of the life sciences strategy and commitment to 5 million genomes, or at least genomic tests, delivered in the next 5 years. And then actually in the last couple of weeks, again, the release of the genomic implementation plan. So really huge amounts of political focus on genomics in the UK. And so a real time to make sure advances in cancer susceptibility genetics are part of that. And of course, I think cancer susceptibility genetics characterises a lot of the challenges evident elsewhere in susceptibility genomics, but often we're a little bit further along as there has been more testing and more data in our genes. And I think, obviously this is more broadly applicable, but it's really important to think about risk on 3 levels or in 3 frameworks. There's the variant level perspective around, is that variant benign or pathogenic, and what is the evidence for it? There's then the data we look at on a gene level. So for a given gene, here's BRCA1 and 2, what are the phenotypes associated with the gene, and what is the absolute risk and what is the age-related risk? But really importantly, we also need to think about, on an intervention level, what is the level of risk reduction or risk mitigation? And again, how does that relate to absolute risk and age-related risk? And of course, something that we are quite simplistic about is thinking that each variant has the same level of risk. And we have for over a decade known that protein truncating mutations in BRCA1 and 2 have different risks of breast cancer and ovarian cancer, depending where in the gene they arise. And thinking across genes, in many of our classic loss of function cancer susceptibility genes, truncating mutations as a group have been shown to have higher risks than pathogenic missense mutations as a group. That's definitely true for BRCA1 and 2. But conversely, there are other genes in which the missense mutations have dramatically higher risk. So certainly the V2424G variant in ATM would appear to have much higher risks than other variants, and particularly the truncating variants. And likewise, as a group, the classic dominant negative missense variants in TP53 seem to have a very higher risk, and particular for the classic Li-Fraumeni phenotype compared to truncating mutations. So we really, this simplistic paradigm of all variants having the same risks, it misrepresents what is likely to be Mother Nature's truth for nearly all genes. And then thinking about the biological complexity, we talk quite simply about risk of breast cancer and risk of ovarian cancer, but of course there are different risks for different cancer subtypes. So we know that BRCA1 has a much higher risk of triple negative cancer, and indeed BARD1, RAD51C, RAD51D seem to be exclusively associated with triple negative breast cancer. And of course, triple negative breast cancer compared to ER positive breast cancer, these are 2 very different beasts in terms of how you treat them, but also their outcome, with very different survival profiles. But of course, thinking about our interventions, our chemoprophylaxis in the form of anti-estrogen therapy is only going to be effective on the risk of ER positive disease. And there are some concerns, in fact, that it may possibly increase the risk of ER negative disease by suppressing estrogen receptor expression in emerging cancers. So a large amount of complexity there about the phenotype as well. Then thinking about kind of epidemiological complexity, we have these age-related risks. And of course, if you think about our interventions, the efficacy of mammography will vary quite dramatically with age because it's related to breast density, and breast density affects not only risk, but masking of the breast cancer that's developed. We also then think about our use of anti-estrogen drugs. What age should they be given? We know the data, the studies have been based on 5 years administration, causing a 40% reduction in risk. But when should it be given? How long does the effect last? And are those side effects of the anti-estrogen therapy, the endometrial cancer, the embolic events, they are quite age-related in their impact as well. So there is a lot to think about here in terms of the epidemiology of the interventions that we have available. We know that the interactions of different risk factors are probably more complex than our simple log linear models suggest. We know there is a changing background risk for different cancers, so breast cancer definitely, and we also know that there are risks related to different ethnicities. We then think about ascertainment bias in terms of our data, that we certainly know that our data on less well characterized genes does not represent the true risks when they're ascertained in the population. And we know that there's likely to be population stratification between different ethnicities. So we put these things together, that is a hell of a lot of complexities around risk that we need to consider in this simple paradigm of testing for cancer susceptibility genes. We then think about the people that need to understand all this risk. We've got our laboratory scientists, our clinical geneticists and our genetic counselors in the old model where we delivered all of this through clinical genetics. But of course, increasingly, as we mainstream delivery of genetic tests, we need to think about the surgeons, the oncologists, the clinical nurse specialists understanding what these risks mean. And of course, the patient themselves, if they are going to make informed choices about the options available. So how do we communicate risk? Do we talk about relative risks, absolute risks? Do we talk about lifetime risks? Do we think about residual risks for a patient of a given age? And of course, I think what's done very poorly is thinking about competing risks. So your risk of comorbidity and your actual life expectancy. For example, if you are talking about risk reducing ovarian surgery for a 70-year-old, a 72-year-old, a 75-year-old, is this different for BRCA1, for BRCA2? So again, there are lots and lots of elements to how we think about and communicate risk. And of course, the consequences if we get it wrong mean that we offer inconsistent management within even a given family. If one is managed in Exeter and their sister is managed in Newcastle, are we managing them equivalently for an equivalent familial risk? We misdirect resource. If we get our risks wrong, then we're misapportioning our resource for prevention and early detection to situations in which there is less impact, and the increasing cloud of medical litigation. So we really, as a community, a group of us were talking about this a few years ago, and thinking about the fact that our model is we have gene discovery. So gene is identified via some manner of exploration in the laboratory. So we find that PALB2 is a breast cancer susceptibility gene. Other labs, other groups characterize this, replicate the experiment, find the frequency of mutations or the spectrum in different disease groups. And then a bit of time ticks by and then we go, oh, yep, we're going to test PALB2 in the clinic. And there's actually a really big gap here, firstly, in all the different types of information that are required to ensure that delivery is safe, robust, and optimal, but also in getting data back from the clinic about what happens when we test these genes in the real world. So we felt we really wanted a program of work to bridge this translational gap, not least on behalf of those at the clinical end, sort of really active monitoring of emerging research and data, but also all those resources that make our clinical implementation safe and effective. So resources about variants and which are pathogenic, clinical guidance over who to test and how to manage them, tools for patients to enable them to make decisions about whether to have the test and what their options are depending on the findings. And of course, educating the breadth of medical practitioners who need to know and understand and juggle different elements and to different degrees, those types of complex risk information I was just describing. And then we really want to link our data back again and ensure that the data from the clinic are being used to inform our schema of understanding about these genes. So we described all these elements and called them different work packages, and took this proposal to a broader group of respective experts in these areas. So people like Jem Rashbash in the Cancer Registry, Antonis, Paul, Mark in Cambridge with their expertise around risk, expert clinicians in developing guidance, and other experts, Diana and Clare in psycho-oncology, Kate and Katie in medical education, Nina and Ingrid around ethics. So bringing this all together, we took this to Cancer Research UK as a programme, and they very kindly agreed to fund it, giving us a Catalyst Award spanning 5 years, and bringing together not just those co-investigators I've just named, but we, in our application and our award, bring in many other partner organisations to help us deliver it. So collaborators, Sue Hill and Ellen Graham from NHS England, Annika Sellers from Health Education England, the Genomic Education Programme, partners in HDR UK, Genomics England, Health Data Insight, various relevant commercial organisations. The professional body is absolutely key to the delivery, particularly the Cancer Genetics Group in the UK, really central to that we worked in partnership with them. Several patient groups who I will come to, but have been really key in forming our PPI group. And then various other bodies, ClinVar and ClinGen, we've had a lot of interaction with, and Enigma and Insight as well. So really trying to bring together and be a hub for this broad ecosystem of partners and bodies important in this area. And so I'm just going to take you through a little bit of what each work package has been up to. So work package 1 was this sort of arrow back from the clinic to the research, so getting the data out of the labs and making it of an appropriate format and utility to be used to further understand these genes. Key to this work package, Jem Rashbash at Public Health England, National Cancer Registration Service, Eva Morris in Leeds, who's an expert in data linkage from cancer registries, and Antonis Antoniou, a real leader in breast cancer epidemiology, also involved Paul, Mark and myself. So the work around linking up our lab data and bringing it back into the research arena predates this programme back to about 2016. John Byrne and Jem Rashbash got together, triggered by the BRCA challenge, the notion of really improving classification of BRCA genes. So they set up the infrastructure by which we could get the data out of the labs in a safe, secure way that met the NHS governance in terms of the Caldecott Guardians and also the Public Health England governance. So because the patient data contains patients who have cancer and patients who don't have cancer, it would not all be covered by our Section 251 privileges that Public Health England hold around data in patients with cancer. So the PHE do not automatically have the right to see de-identified data because not all these patients have cancer. So the data are then submitted from the labs via a pseudonymization process, where a one-way cryptographic hash function is created, bringing together all their identifiable data in a unique pseudo ID. So it sits in Public Health England with the pseudo ID. So this has been set up, and we have data coming in from nearly all of the English labs have now submitted full data, and 2 of the English labs are in process, and we're working with Wales are on board, and potentially we're talking to Northern Ireland and Scotland about getting their data in. And Stephen Hardy has been absolutely key in doing this. What we can then do is collapse the data into summary data, and this can then be exported and I then, via permission from the Office for Data Release, can share the collapsed variant counts, and I'll show you some of those later, back with the labs themselves. But the really interesting research opportunity comes from linking these data, the genomics data, to the different data sources held in the cancer registry. So we've got diagnosis, we've got pathology, we've got recurrence, we've got chemo and radiotherapy, and data on somatic testing. We also in NHS Digital have hospital episodes, death and prescription data. So we can create a longitudinal life course data set for each patient along with their germline genomic data. These data will then all be linked together and led by Eva Morris. These data are going to be moved to a secure research environment led by CRUK, and then led by Antonis. We have 2 PhD students, a postdoc, and 2 clinical fellows doing tester projects working on these data to address questions about second cancers, treatments, outcomes, and so forth. So lots, just getting going, lots of challenges with governance and so forth, but lots of quite exciting potential and a real first, a world first, if we can get all of our data from the labs linked and available for researchers. So really, really exciting and really challenging. The second work package is around resources for variant interpretation, and this is my area of passion. So we're all aware of the ACMG framework and have been working over the last 5 years to implement this in our labs. But there are many, many challenges. So the ACMG gives us structure, but there's huge room there for subjectivity over thresholds and definitions. You have to go to lots of different sources to get your different bits of data. So go to locker-specific databases, go to individual in silico tools. Lots of these resources have a license fees, so Alamet, HGMD. Only a few people in the lab will be able to access those. And it can be very, very time consuming accessing all these different information to classify a variant, and reportedly sometimes taking up to half a day per variant for a laboratory scientist. You need to know your genes and go to different bits of the literature, pulling out different papers, scrolling down through the particular, find table 4 and look up your particular variant. Even nationally, our amassed intelligence on a given variant is spread across the different labs. With different families spread across the country. So trying to pull these together, lots of emailing and checking. And then the labs may have done confirmatory informative assays, for example, an RNA assay on a particular variant, but actually only they hold that information. So again, real problems and inefficiencies across the labs in classifying variants. And there's a lot of anxiety and concern about how we share data and the legality and governance, whether you need to get a patient consent to put data on ClinVar or on Decipher. It's very time consuming checking through the governance and uploading data to different repositories, particularly if you have to check individual consents. And a lot of variable practice from the commercial labs about whether they will or won't share data. And actually for cancer susceptibility genes, it's often a bit more complex because we can't even rely on the sort of de novo occurrence, which gets you lots of points in rare disease. So we're really trying to grapple around case control data, functional assays and phenotypic specificity to classify our genes. With quite loose guidance in the ACMG. So we really wanted to work nationally to improve the situation. So one of our key goals was creating a national laboratory network, create a resource for sharing, some focused analyses to really tell us how to use those evidence items, and to contribute nationally to the international endeavour. So these were the work package 2 objectives. So we started CANVIG UK, Cancer Variant Interpretation Group UK, We have 217 members since starting in 2017. We have had monthly meetings since 2017. We have variant of the month. We develop our consensus specification. We have educational sessions about particular topics. So last month we had a specific focus on high throughput functional assays for TP53. And it's also a forum by which different people on the different international groups can provide updates. So this is our makeup, largely clinical scientists and clinical geneticists in almost equal measure, spread across different expertise within cancer genetics. Some people exclusively in cancer or predominantly in cancer genetics, some people working more broadly. And we had a nice survey, blue is useful, dark blue is very useful, around our membership, around what bits of CANVIG people found useful. Was it the email forum of bouncing around questions throughout the month? Was it variant of the month where people can bring a problematic variant? Did they like the web resource? Did they like updates from international groups? So I think what we're doing is working well, but we will continue to survey our user group. We published just over a year ago our specification, and we're just updating it now and about to release that around the ACMG. So a more precise specification of how to use the ACMG for cancer susceptibility genes. And we also have gene-specific guidance. And then we also published on combining different evidence elements. And again, we've just updated that around which evidence elements can be combined with which rules. In terms of our national resource, this is called CANVAR UK. It's a web resource. So it's got 105 cancer susceptibility genes. So you enter your variant in question, and then it will come up with variant options matching that nomenclature. So you get a front sheet, which tells you live links to CLIMVAR, but also gives some other links out, including allowing you to generate a Google search with some flexible nomenclature for your variant. Lots of in silico tools there. We're really favouring Revel in the UK, but other tools and splicing tools, so all pulled in, different tools that you might need. We also then import specific assays for the genes. So this is a BRCA1 variant. So the relevant assays for that variant, you can go straight there. This is your nomad, and again we present the non-cancer slice of nomad, but you can do females and males and so forth, and it also, we have in there the 1,000 genomes. We then have this data from the labs that I mentioned. So these are the counts. You get a numerator denominator, so you can see here that this variant's been seen 9 times in about 29,000 probands. And then we've got that broken down. We show the white ethnicity probands numerator denominator. We also have here whether the variant's been seen in different locus specific databases. We have here multifactorial assays, so IARC, Eastern, Parsons and so forth. We have different splicing assays for that variant. And we also have splice data submitted from the labs, so that the different labs can just look across and see if another lab has done a splicing assay. And then we have our consensus classification, and you can download a summary sheet. We've done a number of focused analyses. I'll just say, in brief, we did a big analysis of 44 in silico tools against 5 major functional assays. So really quantifying and calibrating those in silico tools and generating likelihood ratios. We've done an assay, an analysis looking at SDH genes and getting what we call a very rare variant missense likelihood ratio. So what does it tell you if you see a single rare variant in a single case with a PHEO? That's an extendable principle, and that's under review at Genetics in Medicine. And then working collaboratively. So we are the first major clinical group in the UK to submit to CLIMVAR. So again, sharing our work internationally. Work package 3, lots of clinical guidance, and we're also having a 2-monthly national MDT for case presentations, which is getting about 150 attendees, so really bringing together the national community. Work package 4, tools for patients. This is led by Clare Foster, Diana Eccles. So they are building a decision aid around risks for different variants and decision making about interventions, building on previous decision aids they have developed, such as breast cancer choices. Medical education, so Katie Snape and Kate Tatton-Brown working with the Winton School in Cambridge for some developing Moodles and also developing a MOOC around risks in cancer susceptibility genes aimed at a breadth of mainstream medical clinicians. And then work package 6, Nina and Ingrid working on some items of ethics, particularly around variant reclassification. So again, in our broader community, we have an expert clinical genetics group, an expert oncology group, an expert primary care group, and an expert laboratory sciences group supporting the various different work streams. We have our PPI group led by Leslie Turner, really supporting particularly those patient tools. We have expert collaborators who we have brought together for our think tanks and our fantastic science advisory committee. Really focused on, at the end of 5 years, having some legacy that we leave behind to allow continuity in terms of our resources, our clinical guidance, our variant data resources, our educational resources, and our linked data sets. So really ensuring that we have places that will allow continuity of these resources. So in the interest of time, I'll finish now, but I'm really keen to answer any questions you have in the question session, and just want to acknowledge all the many, many people who have contributed to the different elements of this programme, and in particular our patient reference panel, Leslie, Helen, Pete and team, who have been absolutely invaluable. So thank you very much for listening. And as I said, I very much look forward to taking any questions.